Yeah, let's start. نحن امبارح حكينا عن the reading frame. قلنا كل messenger RNA محمي باطرافه ب long sequences سميناهم امبارح non-coding sequences، ما بيترجموا. كأنه أنا عندي الأوراق اللي بدي هن يترجموا محميين بغلاف. اوكي هلا هيدول اللونج sequences ما بينعمل لهم translation. They are not translated. Okay, the translation starts from the start codon to a stop codon. From a start to a stop codon. Okay, أنا اليوم حشوف بالضبط كيف عم بتصير عملية translation. Actually, I have 3 steps of translation. Okay, translation is more complicated than transcription. This is why it's not difficult. بس لأنه فيها steps. Okay, عشان هيك أنا بدي إياكم تنتبهوا معي منيح وتركزوا منيح لحتى نلقط الفكرة الأساسية اللي فيها. I have 3 steps. Initiation. To initiate means to start. So it's the beginning, the initiation. The second step is called elongation. And the third step is called termination. Termination, to terminate is to end something. So في يسميهم steps وأنا في يسميهم كمان stages. Steps or stages. هنبلش أكيد بأول step. The initiation. The initiation starts when the small and the large ribosomal subunits, the large and the small ribosomal subunits هن بيكونوا مفترقين عن بعض يا مس، كل وحدة لحال. هيدي الطريقة. So this is the large ribosomal subunit. This is the small ribosomal subunit. They unite together. بيجتمعوا مع بعض وبيصيروا يمشوا على المسنجر RNA. Again, يلا. إذن the ribosome, large والsmall subunit, they unite together. They associate together. They unite together, fuse together. بيصيروا شقفة واحدة وبيصيروا يمشوا على المسنجر RNA. بيمشوا على المسنجر RNA to look for the first AUG and then they settle. بيركزوا. اوكي إذن هن السمول واللارج ribosomal subunits بيجتمعوا مع بعض، بسكروا بطبقوا على المسنجر RNA. They move along the messenger RNA looking as if they are scanning the messenger RNA to look for the first أول AUG بيلتقوا فيها. okay يعني أول start codon. AUG is the start codon. أنا شفتها امبارح بالتيبل. مجرد ما هن لقوها اوكي هيعملوا settlement، يعني رح يعدوا على أو مطرح ال AUG. نحن بنعرف إنه the ribosome, large ribosomal subunit, it has 2 sites. It has the P site, it has the A site. بعدنا بالinitiation. The transfer RNA. أنا عندي كتير transfers RNA. أنا عندي قلت لكم 61 different type of transfer RNA. The one that is specific for AUG يلي هو specific للAUG مفترض يكون عنده anticodon. الA بوجه U، الU بوجه A، والG بوجه C. مفترض يكون عنده anticodon U A C. Okay. AUG if I go and look in the table. نحن عادة table قلت لكم هي للمسنجر RNA، يعني هن الكودونز of the messenger RNA. إذا أنا بروح بشوف بالتيبل ال AUG أي أمينو أسيد حتعطيني، رح لاقي ال AUG بتعطيني the methionine amino acid MET. So من بعد ما الرايبوزوم قعد اوكي على مطرح ال AUG رح يكون جهة ال P site as you can see هي على ال AUG A site رح تكون هون empty. اوكي هيجي transfer RNA هيقعد يلي حكينا عنه هيدا الترانسفير RNA اللي هو specific AUG هيقعد على B site يعمل bind مع AUG bringing the first amino acid اللي هي methionine while the A site is empty. This is initiation. So this is initiation. Let's check it. بيقول the small and the large ribosomal subunits they associate together اجوا مع بعض هيهم. The ribosome now scans the messenger RNA looking for the first AUG. بضل يمشي على المسنجر RNA للاقي أول AUG. بخلي ال AUG تكون على جهة ال P site. اوكي أول سايت عنده أول غرفة عنده. بيقول كمان codon to start the synthesis of protein. The transfer RNA that is specific to AUG الترانسفير RNA اللي هو خاص بال AUG مفترض يكون عنده anticodon UAC ويكون حامل على راسه من فوق MET. اوكي بيقول and carrying the amino acid methionine binds to the P site of ribosome. So this is the initiation. This is the initiation. Is it clear, miss, or you want me to repeat? Is it clear?
Yes.
Okay. Yeah, let's continue. So لحد هلا أنا عندي transfer RNA قاعد بال P site عامل bind مع ال AUG حامل على راسه methionine. The second step of the translation process is the elongation. Elongation, okay? تاني شي بده يصير رح تجي المسنجر رح تجي سوري the transfer RNA هيدي transfer RNA okay that is specific for AGA يلي عندها UCU زي ما شايفينها يلي حاملة على راسها the amino acid يلي هو arginine again arginine بالتيبل هو لل AGA لل messenger RNA اوكي مش للانتيكودون so AGA بالتيبل gives arginine إذن رح يجي يقعد على the A site صار عندي the P site is occupied and the A site is occupied so بالelongation process رح يجي تاني transfer RNA يلي هو specific للsecond codon رح يجي يقعد على بال A site على second codon carrying the second amino acid. تاني شغلة بتصير بالelongation بصير في عنا formation of a peptide bond زي ما شايفينها between these two amino acids. أنا بدي لقطهم للأمينو acids مع بعض. الترانسفير RNA هو بس وسيلة لحتى يعطيني الأمينو أسيد بس أنا اللي بدي إياه آخر شيء at the end I want the peptide بدي البروتين اللي بحصل عليه. فإذن تاني خطوة رح تصير formation of peptide bond between these two amino acids. أول خطوة the second transfer RNA will bind in the A site. تاني step formation of peptide bonds between these two amino acids. ثالث خطوة translocation of ribosome. مجرد ما أنا لقطت اثنين مع بعض the amino acids صار في يمشي الرايبوزوم one more codon في يمشي codon. كيف؟ أنا لاحظوا معي هون كان عندي أنا ال AUG أوكي وال AGA هلأ بده يمشي خطوة واحدة الرايبوزوم بس one codon أنا لقطتهم مع بعض، حافظت عليهم واثنيناتهم ملقطين بهيدا transfer RNA. حيجي هلا the ribosome will move just one codon like this. اوكي، مجرد ما مشي one codon the ribosome، ركزوا معي منيح مس، صار عندي ال A site هو الترانسفير RNA عامل بايند مع الماسنجر RNA. لما أنا مشيت الرايبوزوم، الغرفة اللي كانت هي هيدا الترانسفير RNA كان بال A site صار بال P site. Whereas the A site is empty now, ready to receive another transfer RNA. I will repeat. Just 1 second. Okay, yeah. إذن again أول خطوة ال A site اللي كانت empty حيجي the transfer RNA specific لل AGA binding okay in the A site يلي حامل الأرجينين. This is the first step. The second step أنا بلقط هلا 2 أمينو أسيد مع بعض لحتى أحافظ عليهم إذا خسرت الترانسفير RNA. فإذا أنا بدي أحافظ على هو 2 مع بعض. ثالث step يلي هي translocation يعني movement of ribosomes. The ribosomes will move one codon. اوكي، يعني إذا كانت هي على AUG و AGA هلا بدها تصير على AGA و CUG. هيدي الرايبوزوم. اوكي، هون اللي إلها البي سايت وهيدي الاي سايت. الترانسفير RNA هن ملقطين بالانتي كودون بالمسنجر RNA، هن ارتباطهم مع المسنجر RNA. أنا صار عندي هلا هيدي لحالها فري. اوكي، وهيدي الترانسفير RNA يلي هي ملقطة AGA عندها 2 amino acids the MET and the ARG. حيجي هلا عندي ثالث amino acid as you can see GAC هي انتي كودون CUG حاملة اللوساين. كمان أنا عندي رح يصير عندي peptide bond. برجع بعيد الستبس between these two amino acids. اوكي، هلا بدها تمشي the ribosome one codon. It will move one codon. يعني أنا لازم لعندي CGA. إذا this is the P site and this is the A site. اللي كان عندي بالA site صار بالP site لأنه مشيت الرايبوزوم. P site stands for peptide site يلي هي عندها البيبتايد يلي بيتصمد فيها البيبتايد. A site stands for the amino acid site يلي هي عم بتكون حاملة بس واحد amino acid. هون ليكو بالبي سايت أنا عندي transfer RNA على طول عم بتكون حاملة البيبتايد عم تتصمد عندها. وحيكفي هيدا الشيء. It will be repeated كمان. transfer RNA حيجي يعمل bind A site لأنها empty. بعدين formation of peptide bond between these two amino acids A P site. بعدين translocation of the ribosome. These 3 steps will be repeated until we reach a stop codon. Is it clear, miss? Is it clear?
clear.
Okay. أنا بس عم شوف who joined لأنه أنا أخذت أول شيء attendance. Okay. Yeah, let's continue. So بالelongation بتقول the second transfer RNA اللي هو specific for the next codon اللي in this case AGA اللي عنده UCU as an anticodon and carrying Arg amino acid binds to the B site. Binds to the A site. Sorry. Okay, binds to the A site. A peptide bond is formed between the two amino acids, MET and Arg. The ribosome moves one codon هيدي بنقلها ribosome translocation. هلا ال A site is now empty again and the transfer RNA يلي عنده amino acid okay بيروح على ال P site. Another transfer RNA specific ل CUGU and this continues لحتى أوصل ل one of the three stop codons. Okay, أنا حانتقل هلا للtermination. Termination. Whenever we reach a stop codon, قلت لكم ال stop codons هن one out of the three يلي UAA ما ضروري نحن نحفظهم حيكون معنا التيبل UAG and UGA. So if we reach one out of these three stop codons شوفوا مثلا هون أنا وصلت لل UAG عم بتكفي كانت الالونجيشن شوفوا قد ايه صارت long chain of amino acid اوكي وصلت لل UAG. أنا قلت لكم إنه أنا عندي 61 different types of transfer RNA. اوكي عدد الكودونز that have amino acid. أنا عندي 3 codons ما بيعطوا amino acid هودي ما إلهم transfer RNA. حتنطر for a little time ولا transfer RNA حيجي يعمل bind لل UAG. Okay, so this is the end of the termination of the translation. لأنه ما عندي ولا transfer RNA بيقدر يجي يعمل bind على UAG أو UAA أو UGA. So this is a signal هيدي إشارة to end translation حتى تنهي ال translation. What happens? Okay يلا. What happens? يلا لحظة just 1 second. Okay, so أول شي بيصير الرايبوزوم اللي عملوا association they dissociate بيبعدوا عن بعض. اوكي إذن هيدول large ribosomal subunit and the small ribosomal subunit they dissociate away from each other. تاني شغلة the peptide كمان will be released. So عندي separation of both ribosomal subunits. أنا عندي release of the synthesized protein كمان وأكيد release لtransfer RNA وفي عندي خطوة ثالثة بتصير يلي هي الميثيونين يلي هو بلشت عملية الترانسليشن فيه it will be released. هيدا كان بس signal it's not a part of the protein. So again when we reach the stop codon شو حيصير عندي بالترمينيشن يعني اعمل liberation للبروتين اوكي البروتين راح اتخلص من الميثيونين لانه هو كان بس signal to start عملية translation it's not a part of the protein and the small and the large ribosomal subunits they will dissociate or they will separate. So ribosome reaches a stop codon where no transfer RNA is found to bind on the stop codon. This is a signal for the translation to end. The peptide is released and the large and the small ribosomal subunits dissociate. The first amino acid methionine is removed. Is it clear? Is it clear, miss?
It's clear.
Okay. أنا هأحضر معكم هيدي الرسمة موجودة بالكتاب بنعلمها document D. Okay, it's the step of translation. بنعلمها، حنحضر مع بعض هلا animation. يلا هلا هيدي الانيميشن نحن حضرنا نصها لعند transcription باكتيفيتي 3. اوكي هلا translation and لأوصل لعند البروتين حشوفها. يلا let's watch it together ونرجع نعمل comment عليها.
The process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, But when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported.
Okay, that's enough here.
The process.
Okay, هنشوف animation تانية هتفرجيني بس الميكانيزم. Okay, yeah.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids.
هيدي البولي A tail هي non-coding region يلي بتقدر تحمي the messenger RNA codons. مش قلت لكم ترافا بيكونوا هن non-coding ما بيترجموا بس عم بيحموا الكودونز.
A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA.
شفتوا كيف لما مشي عنا الرايبوزوم صار عندي اللي هو كان A site هذا الترانسفير RNA كان بال A site صار بال P site حامل البيبتايد و a new transfer RNA specific for the new codon come and binds in the A site.
RNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay. Do you have any question, Miss? Do you have any question? Do you have any question about the mechanism? احنا ما حنكتب هيدي السيشن، حنكتب next session. If you have questions, miss, you can ask me now.
لا 11 سؤال.
سارة بدك تسألي شيء؟
هلا قلت نفس الشيء إنه ما حدا عنده سؤال.
Okay. Okay then, if you don't have questions, this is it. أنا ما حأرجع يعني ما حأعطيكم شيء أكتر من الميكانيزم اليوم لأنه موجودين يعني I can continue بس هالقد منيح. You can leave mess وأنا بشوفكم الأسبوع الجاي بنكفي بنفس الأكتيفيتي وبنكتب كل الشرح.
اوكي بس يعطيك العافية.
الله يعافيك.